I'm going to pick gene 3 to walk through together and just kind of demonstrate how we do one of these annotations. So to start out, I need to get a sense of this gene. I can locally blast this genome. If I click on here, I'll end up in this site that has my FASTA file automatically input into the genome sequence text box. I can hit blast, I've already done that, and then I will get from there a list of the phages that are on PhagesDB that are the most similar to Mulch Explorer. Of course, it hits Mulch Explorer as the top hit because it's identical to itself, so let's ignore that one and just look for other phages besides Mulch Explorer that are similar to Mulch Explorer. So we have here Camberlium, QuickMath, Juice 456. All of them and many others have an e-value of 0. That means that when you were searching a database this size that was made of just random sequences instead of actual phage genomes, you would expect to get a hit this good 0 times. So the fact that we are getting a hit this good means this is not random chance. We're actually looking at something that's similar to the Mulch Explorer genome sequence because it has a similar biology. So knowing that we have Camberlium, QuickMath, and Juice 456 as some of our most similar genomes is helpful to us, so it gives us a point of comparison that we can use. If I go over here and pull up the Camberlium page, we can see that it was sequenced in 2015. QuickMath was sequenced in 2017. And Juice 456 was sequenced in 2018. I went ahead and pulled one more from the list, Starterator, as well, that was actually sequenced more recently in 2024, just down the road. And so these are the genomes that I can kind of look at for comparison. I like including a genome that's more recent because sometimes functional annotations, instructions for doing the functional annotations do change over time, so it's nice to have something a little bit more recent. So I'm here on Phamerator, and I'm going to select my genes. This page is slowing down Firefox, I'm sure it is. So I'm going to go ahead and search for F1. And find Mulch Explorer, our draft genome. And I'm going to find our other genomes that we've identified as being similar. Camberlium, Juice, and Phamerator. And QuickMath. Great. So now I'm going to go all the way back to the top, go over and view map, and I can see my different genomes aligned with each other. I can zoom in if I hit down Control or Command, depending on whether or not you're on a Mac or a PC. I can bring Mulch Explorer up to the top by just clicking on its name and pulling it up to the top if I want to look at it there. And I want to look at gene 3. That's the gene that I'm going to be annotating. So we have it here. Starterator is actually quite different, it looks like. So maybe I'll pull him down. Maybe it's not as useful to us for this particular gene. So if I kind of zoom in on, not quite that much, on gene 3, I can see in some of these other genomes, Juice 456 and Camberlium, that the gene 3 is a terminase large terminase subunit. We can see already that the names that are being given for the functional annotation are not 100% match across the board. So we're going to have to be very careful to follow the instructions once we get to functional annotations to follow the instructions for whatever the most updated version of the name of this gene is. And we can see that this gene is present in a lot of similar genomes. So that just kind of gives us our bearing for what we're looking at. But now to get started with the annotation, we have a couple questions. One, do we think it's a real gene? Just the fact that we see it here in these other genomes that have already been annotated is sort of a starting point for us to say, well, people thought it was a real gene over there, so might still be a real gene here. We'll have to double check the coding potential when we're looking at the start sites, but for right now I think we can say, alright, it looks like we're looking at a real gene. Let's try to figure out which of the possible start sites is the start site that we want to use. So in order to do that, I'm going to come over to DNA Master. I'm going to go ahead and widen this feature list just to make it easier to look at both the start and stop coordinates here. This is our gene that we're working on, that gene 3. I really want to use that frames window to give me a sense of where the genes are. So I'm going to go to DNA Frames. And I'm going to bring all of the genes that have been annotated over here into this window. And I'm going to zoom in and on gene 3. I can navigate to the left and right over here, zoom in and out. I'm going to zoom in even more. What I really want is just to see gene 3 and the gene to the left and right of it. I don't really need to see anything else. So I have all of the stop site that is stopping this gene is right here. So that's going to end that stop codon. The other end of the open reading frame is right here. There's a stop codon, so the gene can't be any longer than this. And the short lines all the way across are potential start sites that we have to pick between. Now, generally speaking, you're going to pick one of the earlier start sites because we have all of this great open reading frame, this region that's clear from stop codons. We don't want to waste that by starting the gene somewhere in the middle. We're going to use all of this clear and open reading frame and kind of take advantage of that to make our protein. But of course, the first start site is not always the correct start site. Very often it isn't. So we have to distinguish which of these start sites are we going to select. But you can generally assume it's probably going to be one of the first few, maybe 5, at least 10 start sites in the gene is a pretty fair assumption. So we can already start to see that our gene is looking like, as it's auto-annotated, it overlaps with gene 2 a little bit. We see that the auto-annotation is not using the first of the possible available start sites. We have one earlier that we could pick that would give us even more overlap with gene 2. And we could pick a later start site that would give us no overlap, and it would give us a bit of a gap between genes 2 and 3. So that kind of gives us our bearing of what we're looking at. And if we want to see what those start sites are, what coordinates go with each of those start sites, we can open up that RBS window. And here at the RBS window, we see that our very first start site is is affiliated with a TTG start codon at position 659 in the genome. And then the second one is an ATG at position 671, another ATG. So that kind of covers those first 3 that we just highlighted. 1, 2, 3. So we do want to make a note that one of our early start codons is a TTG. That's going to be becoming important when we look at coding potential, so we can identify where that is in our GeneMarkS coding potential graph. But we also want to make sure that we're looking at the same gene as the gene that we're looking at in Phamerator. So, in order to know that we're looking at the same gene, we have to look at the stop codon. So the stop codon, or the stop site within the stop codon, the last nucleotide of the stop codon, is 2,158 base pairs into the Mulch Explorer genome. Another way we could find that is over here. We could see our gene 3 starts 671 base pairs into the genome and stops 2,158 base pairs into the genome. So we want to compare that over here with the online version of the gene in Phamerator. I'll click on that, and I can see that their auto annotation says that it starts at 671, stops at 2158. So the same stop codon means that we're looking at the same gene. They picked 671 as their start. That's the same start site that was auto-annotated in DNA Master. These are two different auto-annotations. The people who are hosting this online have done an auto-annotation through the Phages program. We have done our own on our own desktops, and they can be slightly different from each other. So we have to check that we can't just go by the gene number and say that we're looking at the same gene online versus on DNA Master. We have to go with the stop coordinate, and we can also compare if they got the same start site as their auto-annotated start site. So it looks like everything's matching up. That makes it easier for us to kind of work through and compare information from different places. So the most important piece of data that we need to look at is the coding potential. We're going to be looking at the GeneMarkS coding potential. So that's the coding potential that was trained on itself, on its own genome, on Mulch Explorer's genome itself. Basically, GeneMarkS is looking for long open reading frames, and it's saying if there's a long open reading frame, we can assume that there's a real gene somewhere in that open reading frame. And it's looking for sequences that stand out as matching that, or matching a real gene, as opposed to the kind of sequences that you would have in a sequence that's not encoding a real gene, or in a different frame that's not encoding a real gene. So this is the most important piece of data that we can use to answer two questions. One, are we looking at a real gene? And two, where do we expect the gene to start in terms of where is it translated? Where does the protein that is translated from this gene begin? So we have to find where in this GeneMarkS graph we want to look. We're going to be looking primarily at the black line. The black line is order 4 coding potential. That means the software took 4 nucleotides at a time to look for these patterns of sequence that are more common in real genes than in places of the genome that don't have a protein coding genes. Order 2 coding potential, we mostly ignore. The software is using just 2 nucleotides or 2 base pairs at a time in order to get these signals of what patterns are more commonly associated with real genes than in or real protein coding genes than areas that don't encode protein. So it's really useful to be able to compare this GeneMarkS graph with the list of possible real start sites over here from DNA Master. So I like to be able to have those side by side when possible. So I could try to start to find where is my gene in this, and where are the start sites that I can see over here in my RBS window. How do I find them over here in the GeneMarkS graph? So first thing I need to do is orient myself in this GeneMarkS graph. Basically, these top 3 rows are the first 3 reading frames. They're all forward reading frames. These bottom 3 rows are all reverse reading frames, and the graph kind of ends right here at just after 2000. And if I go in the second row right here is what I'm highlighting here. So if I wanted to pick up in that same reading frame, I'd have to go to the second row of the next page to pick up where I left off. So you could basically take one page and copy and paste it to the right of the previous page, and you would get maybe a more intuitive sense of how this is all put together. So I need to find my start and stop coordinates, particularly my start coordinate on this graph. So I'm looking for a start site, let's start with the ATG. So I'm looking for this. This is the auto-annotated start site, this ATG, and it should be at position 671. So I need to find position 671 on this graph. So all of these tick marks, this tick mark, this tick mark, this tick mark, this tick mark, each of those represents 100 base pairs. So if this is 400, then this is 500 and 600. Of course, 671 is going to be about 71% between 600 and 700. So we're looking about 70% of the way between here and here. So this would be right about where I would expect to find that ATG start site here. Sure enough, there is a start site labeled right here, exactly where I expected to find it. So we know that this second reading frame is the reading frame that we're looking at for our gene number 3. Just to point out, these longer lines, these longer tick marks are ATGs. The shorter tick marks, you can see one right here, those are GTGs. And the TTG start sites are actually not drawn, or they're invisible on this graph. So that's something you have to be really careful about when we're using the GeneMarkS graph. We have to make sure that we're comparing the this list of possible start sites with the GeneMarkS graph so that we're not ignoring any TTGs. So we have this TTG that's one of the possible first start sites, so we're going to have to manually kind of figure out where we expect that one. It's not drawn in for us. So it's going to be at 659, position 659. So again, that's going to be 400, 500, 600, and 59% of the way between 600 and 700. So pretty close. This was 59, 69, and the next one is 71. So we're only looking about 12 base pairs different. So we're looking probably just before the ATG that we found. We can throw in this TTG. So that gives us a perspective of where we're looking for that start codon. And then if we look at the third possible start site, another ATG, we can find that right about here there's another ATG, so I'm expecting that that's going to be my third start site, but let's just double check based on the position. 797, so 700 is right here, 97 is very close to 800, so we're looking right just before 800. If I go up, that's exactly where I expected to find it. That ATG is right where we expected. So we can kind of tell what we're looking at in this coding potential graph. Does this work? I've never actually drawn on this before. Let's try this. So we have our first is kind of invisible, our second is right here, and our third is right here. This is very hard to draw with, but you'll get the idea. So we have to kind of decide probably between these first 3 start sites during our annotation. Now, of course, this coding potential is the most important piece of information that we're using to determine our correct start codon. And so how are we going to try to figure out what is potentially the best start site using this information? So now's the time where we're kind of oriented to the gene, we have everything pulled up. I'd like to transition over to filling out our entry. So what I'm going to go ahead and do is I have this entry copied from the template in the shared drive. We're going to do a start site annotation, so I'm going to go ahead and copy the template to have a copy of it that I can play around with and use for the annotation. So I'm going to create a copy. Okay. Now I'm going to rename this copy, and this is the third gene, so I'll label it that way. And let's go ahead and start working our way through. So gene number is 3, also 3 in Phamerator. Name of the reviewer, that would be me. And the way we're going to work through this is we're going to work through the rows that are labeled in blue, and then we're going to work all the way to the bottom, only answering the questions in the rows that are labeled in blue. And then we're going to go back a second time and just answer all the rest of the questions, the ones in red. So basically all the questions that are labeled in red are asking about something where you need to know which start site you end up picking as being the correct one before you can answer the question. And so, of course, we can't start by knowing which one is the correct one. We have to gather data first. So we're going to do all this data gathering first, make our decision, and then come back and fill out the information that kind of builds the argument in defense of the start site that we end up picking. So let's go skip down to this next blue question. What is the stop coordinate for this gene? We've already pulled that up. Our stop is 2158. 58. Next blue question, auto-annotated start sites in DNA master. Start site chosen by GLIMR. And then the next one is start site chosen by GeneMark. Best way to interpret this, if you're a little stuck, is to come over here, double click on this instruction cell, and it goes over how to read the output. If DNA master only tells you what GLIMR decided, that means that GLIMR and GeneMark both agree. If it only tells you the output from GeneMark, it means that GLIMR didn't even come up with this as being a real gene. And if it tells you separately what GLIMR and GeneMark decided, that means that GLIMR and GeneMark disagreed, and so that you can kind of write down both of those auto annotations separately. So let's go over and see what was auto annotated in GLIMR and GeneMark based on DNA master. So I'm here, I've clicked on gene number 3, and it says GLIMR has called base pair 671. So if it just says what GLIMR has called, that means that both GLIMR and GeneMark have both called the same thing, so both of them have called 671. So GLIMR and GeneMark are using the same information that we're looking at when we look at this graph right here. Where does the coding potential spike relative to the positions of these start sites? I think it's also able to look a little bit at sequences like RBS sequences and things like that. I think that is also included somewhat in that analysis. So 671, we can write that in for both. And our we can go down to the next blue question, coding potential. Open reading frame has coding potential according to GeneMarkS. So that's a clear yes. We can see that there is coding potential, quite a bit of it, in the region where we expect this gene to be. And so yes, this looks like a real gene. It has real coding potential. So yeah, great. We also need to know what coding potential tells us about which start site is probably the best start site, or at least best supported by the GeneMarkS graph. So in order to do that, we're going to go back to what we had just looked at, our numbered start sites, the invisible TTG, the ATG, and the second ATG. And what we're looking for is we want to pick a start site that encapsulates all of the coding potential. So that means we basically want a start site that starts to the left of our big spike in coding potential, but just barely to the left. The reason for that is once the coding potential spikes, that's evidence that all of the area where that has high coding potential is really a part of a real protein coding gene. So if I think that this area is really a part of a real protein coding gene, the area I'm circling here with my mouse, then that means that if I picked this as my start site, I would be cutting off all of the coding potential upstream of the start site and saying that it's not part of a real protein coding gene. But since it has that high coding potential, we want it to be included in the gene that we call, and we want our start site to be to the left of it so that all that coding potential is part of the gene and not sitting on the outside of what we think is the real length of the gene. So just going by this, I think we can say that 3 is pretty well ruled out. That third start site is pretty well ruled out because it cuts out some of this beautiful coding potential. We don't want to cut that out. So over here, start sites 1 and 2, there's a reasonable amount of space here between the start site and the peak in coding potential. So I would say whichever one is closest to this peak in coding potential would maybe be like the best supported by this graph. But certainly, I mean, these two are very close to each other, 1 and 2, so you couldn't necessarily eliminate one versus the other just based on this, based on their position, because they're so close to each other. So I would say if I had to make a conclusion from this, I would say the best supported start site based off of this graph is the second one, followed by the first one, and we can pretty well eliminate the third and everything after that. So, start site best supported by the GeneMarkS graph would be the second one at 671. I'll go ahead and write that. 671, and here's a place can't eliminate. What's the first one? 659. 659. Not what's the third one? 797. 797 or later. Okay. Additional observations from GeneMark host, optional. So that would mean if we're getting kind of confusing information from this, we could go back and run GeneMark smegmatis. Mycobacterium smegmatis is our host for Mulch Explorer. So GeneMarkS was trained on the smegmatis genome to look for what are nucleotide sequences that are associated with real protein coding genes in Mycobacterium smegmatis. And it can give you sort of similar results, but they can look different. So it can give you just a different angle or a different perspective on this coding potential data. Not an obligation to look at that, but when things start to get weird, it can be an interesting thing to pull out. So screenshot of GeneMarkS data. I'm going to open up the snipping tool here and navigate back to the coding potential data. Let me zoom out so I can see at least 3 of these reading frames. I can get a sense of which reading frame we're looking at. And I'm going to take a screenshot, including at least part of the previous gene so I can see where it is relative to the upstream gene. I'm going to get 3 reading frames in here. So I'll go ahead and paste my image, put it in there so I can have it for future use. Alright, so that allows me to go on to the next section. That's the coding potential. Again, that's the most important part, but I also need to kind of give a sense of where the gene is relative to other things in the genome. So for one thing, I want to know, is this a gene that's going in the forward direction or a gene that's going in the reverse direction? I probably would honestly know this before I ever get to looking at the coding potential. But here we have, I just put the coding potential first because it's so important. But here we can see that the gene is drawn above the little ruler. So that means that the gene is in the forward direction. If the gene was drawn below the little ruler, like this gene here, that would mean that the gene was encoded in the reverse direction. I could also figure out the same information by looking at the frames window. If I get back to here, I can see that these first 3 top 3 reading frames are the forward reading frames, the bottom 3 are the reverse. You can also tell that the forward genes are shown in green, and the reverse genes are shown in red. So many different ways to figure out if you're looking at a forward versus a reverse gene. Alright, so I'm looking at a forward gene, and where does the upstream gene end? Alright, so by end, assuming that my gene is a forward gene, the upstream gene is also a forward gene, end is going to mean where's the stop codon of the upstream gene. If I was looking at a reverse gene, so that my start codon would be here, my stop codon would be here, my upstream gene would be now to the right, and this gene, 18, is in the forward direction, So where does this gene end? By that what I mean is where's the start codon there. So it's just how big is the gap between the start codon, we're trying to figure out the gap between the start codon of your gene and wherever you start getting part of the upstream gene. So I'm trying not to use words like where's the start codon or stop codon of the upstream gene, but just where does it end? Where does the part that's closest to the part we're interested in stop? So a great way to do this is just that main menu, the features list in DNA Master. If I'm looking at gene number 3, which that's in the forward direction, my upstream gene, the one closest to the start site and furthest from the stop site, is going to be gene number 2. Gene number 2 starts at 445 and ends at 699. So that would be where my upstream gene ends, 699. So now I need to figure out the gap between where my upstream gene ends and my auto-annotated start codon. So the obvious way to do this is to start by subtracting this number from where my auto-annotated start site, which is 671. So if I were to take, I'll just pull out a calculator here. We can do 671 minus where 699. So we're getting a negative number here. So we actually know that the end of gene number 2 and the beginning of gene number 3 overlap with each other. So you can kind of see it right here that this overlaps. You can see it in the frames window of DNA Master, that the end of gene 2 and the beginning of gene 3 are overlapping, assuming that you use this auto-annotated start site. And we've pretty much eliminated the third start codon as being the correct one because it cuts off too much of the coding potential. So we're really just trying to decide between these two, this TTG and this ATG. Now, in my mind, once you start to have an overlap, you're going to try to minimize the overlap if you can. So there's no good reason to pick the earlier start site and just make that overlap a little bit longer than it needs to be. So this gap analysis, I think, is going to point us in the direction of picking the second of the two start sites. Now, it's not quite as simple as being able to just subtract because you actually have to count both ends. It's kind of like bookends. If we're having the overlap, this nucleotide, the first nucleotide in our start codon, and over here, the last nucleotide in our stop codon, both of these need to count in how much we're overlapping these two genes. So these two genes are going to overlap by 28 plus 1 by 29 base pairs. When the gaps are larger, if you're off by 1 and you're just doing a simple subtraction, I'm not too worried about that. But it does matter when we have those 1 or 4 base pair overlaps to get it precise, because then we know if this is one of those cases where the ribosome can just jump from the stop codon to the start codon of the next gene. But for this, if you were to have written a 28 base pair overlap, I wouldn't look at that too askance. So our next question, if the upstream gene is in the same direction, which yes, our upstream gene number 2 is in the same direction as our gene, they're both forward direction. So if they're in the same direction, are there any start sites that would lead to a 1 base pair or 4 base pair overlap? So gene 2 stops at 699. None of the possible start sites, let me pull up the possible start sites over here in the RBS window, none of them are close to 699. 659, 679, and then 797. None of them are really any close to that 699 where the upstream gene ends. we would be looking for either a start position that was 699 exactly, or we could be looking for a start position that was at 696. That would be a 4 base pair overlap. Both of those would be really strong start sites if they had that 1 or 4 base pair overlap, but no, does not exist here. So moving on. So next up, we need to get a screenshot of the frames window. All right, so let's go back to DNA master, and our frames window will open by, again, DNA frames. I already have it open over here. And so I have gene 3. Let me make this a little bit bigger so I can get a good screenshot. What I want to do is be able to see a little bit of the gene on either side, so a little bit of gene 2, a little bit of gene 4, so that I can see it in context. And I also want to get the full 6 reading frames. That way I can see if there are any genes that are conflicting in here. I can get a context of where we are in the genome. I want to make sure that I get the numbers on the bottom so that I know where we are in the genome. So I'll copy that and paste that over in my template. And alright, next up is the position of the first start codon in the open reading frame. So for that I need to come back to my RBS window. Let me reopen this, I'm not sure I'm looking at the right thing here. There we go. And so of course the position of the very first start codon is 659. So why do I want to get that information? Historically, originally when people were annotating genomes, sometimes it was the default just to pick the first start codon as the correct start codon because people didn't have access at that point to other information like the coding potential. Now we have better ways of annotating, and so we know that certainly it's not always the first start site that's the best. Many times it might not be. But if we think back to what we talked about, where on average you're going to hit a stop codon every 21.3 codons, that means that if you're getting these long stretches of sequence that doesn't have any stop codons, the fact that you have a long open reading frame is in and of itself evidence that you have a gene in that location. And so, again, same way we don't want to waste coding potential, but not quite to that extent, it's a little bit unlikely to cut off significant portions of an open reading frame where you have this statistically unusual low amount of stop codons, or a region free from stop codons. So just as sort of a candidate, that gives us a reference point to know what is our first start codon, and I've already forgotten the number here, 659. Yeah. So does the position of the first start codon in the open reading frame overlap with the coding region of the gene upstream? In our case, yes, this start codon overlaps with gene 2. I could do this visually because it's a pretty clear overlap. I could also do this numerically. So if I went to the stop site of gene 2, 699, and I compare that to the position of this first start codon, clearly 699 comes after 659. So yes, there's an overlap. Yes, and now we need to know by how much. So that would be about 40 base pairs. Now right away this sets off alarm bells because we know from our guiding principles that you can have overlaps between genes, but you really shouldn't have overlaps greater than 30 base pairs. So this on its own is going to help us eliminate that first start codon because you don't want to have an overlap over 30 base pairs. So in general, the way that I would think of it is you want to minimize the amount of overlap that you have. So if you have an option to pick a start site that's a little bit later, and that still makes sense with coding potential and other data, and it allows you to minimize how much your gene is overlapping with the upstream gene, that would potentially be a preferred start site with respect to its position to the upstream gene. Alright, so that finishes that section out. Blast P, this section is listed in black. It basically means I'm somewhat ambivalent of whether or not you go through and fill out this information in your first go through or in your second go through after you've picked your final manually annotated start site. I'm going to go ahead and skip over it for now so that we can focus on the pieces of information that are really going to help us narrow down the correct start site. So that brings us to the Starterator section. We need to pull up our Starterator report. So in order to do that, we first need to make sure that we know what number our gene is in GeneMarkS and Phamerator. We already confirmed that our gene is the same number in GeneMarkS and Phamerator because it has the same stop codon. So we can go ahead and select gene number 3 here. Great. So while we're here at this page, we can just scroll down a bit and take a look at some of the other genes in this fam and how they've been annotated. This is giving us a little hint preview at the functional annotation. We see some that are labeled terminase, some terminase large subunit, so that the functional names don't all agree here. That's kind of a sign that we're going to need to pay extra attention to what the instructions are and what the approved function names are currently. But what we really want right now is the Starterator report. I can click on that, and this brings us to this rather messy looking file. We've got quite a number of genes in this fam, so this file goes on for a long time. The way that I search through it, because it's got 278 pages, so we need a strategy to read this effectively. I usually do Control F and I will search our genome. And that will allow us to determine which track our phage is in. So in this case, our first instance, when we do Control Find, we have 6 hits. The first hit we won't always have, depending on whether or not Mulch Explorer is the genome that's featured in this track label, but we'll always have at least 5 matches. So if I go to the second one, which might be your first one, depending on whether or not you hit something in that image, we can find that our Mulch Explorer gene is in track 400. We've got a lot of tracks here, so track 400. So we can go and we can find track 400, and even if we didn't see Mulch Explorer here, we would know that that's where our genome is represented. So how do we read this image? What are we looking for? We know that we need to find a visual assessment of what we're looking at, so we know how to read this image. So in our case, one thing that you want to pay attention to when you're doing the visual assessment is the background highlighting color. So here you can see a number of tracks that are background highlighted purple. Then down here you can see highlighted pink, purple again, pink again, purple again. So the color alternates. Every time the color alternates, that you know that you're switching to a different subcluster. So if we know that Mulch Explorer is in the F1 subcluster, that means that all of these other phages and all of these other tracks that have the pink labeling, so down to here, we know that those are also going to be phages in subcluster F1. So what we want to look at for the visual assessment is really just look at phages in our same subcluster, especially if we have a large file where we really can't look at everything at once, and try to get a sense of which start sites are conserved within this subcluster. So let's zoom in a bit so we can see a little better what we're looking at. What we want to know is, all right, we have this start site here, 131, this one, 154. We've got one that's a little hard to read, 224 over here. We just want to see, are we seeing those consistently throughout the subcluster? So for 131, I see it. Yes, 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 yes. All the way down. I don't see any that don't have 131. So let's look at 154. Yes, yes, yes. Yes, 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 yes. Likewise, I don't see any that don't have 154. So we've got a lot of conservation of all of these different start sites. We look down here, I don't know the numbers for these, hard to read, but again, they seem pretty well conserved. So what that means is we're not going to be able to distinguish between start sites or find one start site that's better than the other just by looking at the conservation of these start sites. Again, we want to be very careful when we're using a Starterator report to distinguish in our minds between the concept of consensus, human consensus, and biological conservation. Human consensus means that humans have agreed on a particular start site, have selected a particular start site consistently, whereas biological conservation means that your start site is present either across the fam or at least across the members of the fam that are from genomes in the same subcluster. In this particular case, we have basically 100% conservation for the genes in this fam in genomes in this subcluster, so that's not going to help us to distinguish between start sites. Now we do see that there is a consensus. So the green line means that this was picked as the manually annotated start site, so this was selected as the correct start site. By others, if you have yellow, it means that the genome in this track is a draft genome. So it was auto-annotated to be at this start site, but we don't want to put too much confidence behind it because humans haven't looked at that yet. Any other color means it was not selected, not auto-annotated. So you can see a few examples right here where this third start site was called or manually annotated instead of the second. But I would say that the majority of the tracks are using the second start site and calling that the correct start site. So we can put that all of that information over here. Start by just copying the URL. And then it was track 400 that Mulch Explorer was in. And we just talked through our visual assessment. We said that all of the early start sites are conserved in the F1 subcluster. The consensus in the cluster is for the second start site, third selected subtypes. Okay, so that's just my description of what I could get, the information I can get by visually looking at this image. So, Starterator summary of most called annotation in FAM. So again, the way that I like to go through the Starterator report is by using this control find method to find the different instances, the 5 or 6 different instances of our genome's name. So we've talked through the first 2. That helps us interpret the image. And the third one starts to give us some statistics about what we're looking at. So once I get to that third hit, I'm going to scroll up to the header here so we can kind of get a sense of what we're looking at. This category, or this section, is looking at a summary of the final annotations. And really this one sentence is that summary. The summary is of all of the possible start sites that are listed here, the one that was selected most as the correct start site was the one numbered 164. And it was called in 155 out of the 1,226 non-draft or manually annotated genomes. So that's actually not that much, right? There's still many genomes or genes that have a different start site picked, but that would be the most common. So now we need to figure out, does Mulch Explorer have 155 as a start site that's even present in the gene? And if so, did it call 155 as being the correct start site? So we have 3 different categories in this section, and all of our genes are going to be categorized into one of these 3 categories. The first category is, yes, our gene has the start site that is the most annotated, and that is the start site that was selected. The second category is, yes, our gene has the most annotated start site in it, but that's actually not the start site that we picked for this gene. And the third category is, we don't even have that as an option. That start site does not exist in this particular gene. So if we take a look at which category our Mulch Explorer is in, just scroll up to the category header. We can see that our gene fits into the category of genes that do not have the most annotated start. So it doesn't really matter that the most annotated start is 164 for our purposes, because we know that 164 cannot be the correct start site for our gene. So I'm just going to go ahead and just say not in our genome rather than, or our gene, rather than copy in that statement, because it doesn't really help us to know that there's some super common start site out there if we don't even have an option of calling that one. Starterator statement by start number of the most called start. Let's just go ahead and say not in our gene. But what we would be looking for if we were able to, if we had 164 in our genome, is we'd be going down to this fourth or third, depending on how many you have total, category of times where you have the word Mulch Explorer. So this section, if I was to scroll up to the top of this section, is listing each of the possible start sites for all of the genes in this fam, and it's giving us a summary for that particular start site. So this is summary by start number. These summaries are going to go into this category. Summary by start number. So we know that we're interested in the second possible start site. That's the one that we think is the most likely. So the number associated with that one is 154. Now, how do you figure out what 154 actually means? The 154 is just a placeholder number. It doesn't mean anything biological. You can't use it for anything. So how do we translate that 154 number into something that actually is usable? The way that we would do that is by going down to one of the last 2 hits of the word Mulch Explorer and looking for that start number. So 154 is the start number we're looking for, and that is at base pair 671. No surprise there. When we look at what is our second start site, that's 671. Great. So what do we need to know next? Starter position of the most called start site in our genome. Not in our gene. So that finishes out our first Starterator report. Basically, the information that we're trying to get really just came from this visual assessment. In this case, the summaries were not that informative for us. So let's move on to the RBS binding site. This puts us back in DNA master. And we need to look at this RBS window in order to answer these questions. So we need to list out the start sites with a z-score above 2. So our z-score is called our z-value over here. And we only need to list the ones, say, in the first 5 or first 10 possible start sites. After that, the chances that any of the start sites are actually our real ones is pretty low. So in this case, we have 2 start sites in the first 10 possible that have a z-score over 2. So I'm just going to list what those are. So it's the second one, 671, and the third one, 797. So remember that z-score is telling us the likelihood that this sequence, the sequence just to the left of the start codon, it's telling us the likelihood that that sequence contains a Shine-Dalgarno sequence. And if it does contain a Shine-Dalgarno sequence, that increases the likelihood that what you're looking at, what follows, is a real start codon, because the ribosome is going to get recruited to that area. So this would be a piece of evidence in support for either of these 2 start sites. It doesn't really help us distinguish between them too much. They both have a good z-score. But we're going to, in order to make our decision between those 2, we're probably going to have to look at other pieces of data. In this case, the gap analysis is going to help us decide between those 2. So we just also need a screenshot of this window. So I'll go ahead and grab that, paste it in. So you can just take maybe the first 10 possible start sites. You don't need all of them. Great. So that brings us to the end of our first run through, where we work through all of the blue questions. And by this point, ideally, we should be able to figure out which start site is best supported. I think for ours, it's fairly clear that the 671 is the best supported. So I'm going to go ahead and put 671 here. So now it's time to figure out which of the start sites is the best supported using the evidence that we've collected. So I think in our case it's pretty clear that the best supported start site is 671, that second start site that we're using. I have high confidence in this call because we can eliminate the third start site by the coding potential, and we can eliminate the first one by the overlap. And so we're really just by power of elimination, we have the 2 left. So I would say, just write that out, can eliminate third by the because it cuts off too much coding potential, can eliminate second because it makes a too large of an overlap. The second start site is good with respect to coding potential, conservation of the start site, consensus within F1 subcluster, not overall, but within that subcluster, and a good z-score. What I really want to emphasize in going through this is that we're not just getting information about whatever the auto-annotated start site was. We really want to use all of these different pieces of evidence, the coding potential, the overlap analysis, the Starterator report, the RBS sites. We want to compare those pieces of evidence across the different options for possible start sites. We don't just want to make a list of fun facts about one start site. We want to, for each category of evidence, compare the different candidates to each other and say, all right, for this piece of evidence best supports this start site. For this other piece of evidence, this is the best supported start site, and make those comparisons between the possible start sites. It's not just a list of facts about one start site, it's different categories of comparison between the top candidates. All right. We've basically pretty much done the hard labor of figuring out what we think our gene is, and from there we can kind of move on and re-go through and answer all the red questions to kind of build out our argument. So I'm going to pause there.